thank you for being here. The talk today is meant to be, inclusive, a bit more inclusive of all Nemaline varieties. And so we want to actually give, voice and some opportunity for each of the different subtypes to be able to kind of talk through a little bit of, understand a little bit of the research behind each of the different Nemaline subtypes, and then actually understand a bit further about the research that's happening. So we're lucky enough to have 3 primary researchers in Nemaline myopathy here today. So we're going with our theme of amplifying the NM community voice. The tools used to study the spectrum of Nemaline myopathy. All right, Kate, why don't we start? We can go to the next slide and do a little bit of housekeeping. All right, so just if anyone has the need, there's closed captioning available. Should be a clickable link, possibly on the top of your screen. This webinar will be recorded and shared on open platforms. Please, we're not managing mutes today, so please keep yourself muted unless you're called on or you have a question down later in the discussion. So we're trying to have a little bit more conversation today about the different genetic varieties that exist within Nemaline myopathy. And so if you hover over my face on the right-hand side of your screen, you probably can see that I've labeled that I have a child representing the actin gene. And so I put that little label in my name. If you'd like, you can hover over your name with your mouse, click on the 3 little dots, and you can change your name by renaming and just add the little initials of whatever genetic subtype you might present with. And this way we can kind of have that label later, which we're going to go into breakout rooms with different researchers and different AFBS representatives to hear from each different subtype that we can represent. In the chat, Lynn labeled out, how to do that as well. So today's talk is really meant to, attempt to be, an inclusive talk where we're talking about some of the projects that AFBS is actually, representing. And so we want to make sure that in a perfect world, we would be funding projects that serve the entire Nemaline community. And, we've yet to find the perfect project, right? The one that can solve all of this thing for anyone and everyone across age spans, across subtypes. And so as we continue our search for work, some of the researchers today are going to present to you some of the success stories and some of the barriers to those projects. And so we look forward to hearing from all of them. These webinars have taught us a lot. We've learned a little bit more about how important it is to have our community signed up on a single registry. And if we can get everyone signed up, actually that represents more of our community and our numbers, so that when we go to pharma, we will be able to actually represent wholly the pharmaceutical need that exists out there. And there may actually be more demand and more finances to support the work and projects. So registration is important. We use the CMDIR as our registration tool. We also learned about the tissue repository, that if you're having a procedure or surgeries and you are interested in sharing some of the tissues, which we will help organize for you. Stacy Cosette is on this call. I believe she'll put in the chat and her contact information here. And so if you have an upcoming surgery or you have the opportunity to give a little bit of tissue or even a blood sample, we can arrange that for you, do all of the work, get the forms out of the way, and handle the shipping with the medical office themselves. So we're going to take all the burden away and allow you to kind of store those tissues. The tissue repository is important also, not only to have a single time intervention. If you happen to have 3 or 4 years later a second opportunity, the tissues change over time, and so the researchers would like to see that as well. So the tissue repository, the CMDIR are absolutely important. Getting genetic confirmation is also important, and we're working on streamlining that process for anyone in the community as well. We'd love to be able to confirm our numbers so that we're, again, really well represented when it comes to, you know, our talks with pharma. One of our webinars coming up is going to be trying to prepare and plan for a call with the FDA. And so just trying to really move and put ourselves in position to be able to be clinical trial ready. One of those steps this year that we're hoping to engage in is a natural history study. And the more people we have in our community to reach out to and connect with would be amazing. We'll be able to kind of get people tested. That will be support the community and the foundation and its work towards, you know, finding treatments for Nemaline myopathy. Kate, that's cool. Thanks for bouncing out of the slide deck, but I think we can jump back in now. I think in general terms, a lot we've learned from these webinars. Really appreciate the fact that we've got probably one of our higher attendances for this webinar. And we're going to have you know, research updates, you know, models that have been created to use for gene-specific research from Dr. Gupta and Dr. Gonorazky. And then Dr. Granzier has also joined us today, the late add-on since we edited this slide. We appreciate him being here. Gene-specific breakout groups. We're going to have a little bit of breakout groups later, trying to put gene-specific people into those categories. So renaming yourself helps. You'll have the opportunity later to actually select that breakout group, you know, as we give you the opportunity to go into a breakout group later. And then Kate will actually help us by sending messages to the group, letting us know how much longer time frame we have, and then bringing us back to the main group toward the ends for a wrap and any other Q&A discussions. Okay, so here are today's speakers. Dr. Granzier, who's been at the University of Arizona for some time, and he was one of the first researchers that I engaged with as many as 15, 16 years ago. His work in the area of the Neb mouse and the muscle fibers to understand the actual mechanics of the muscle fibers themselves. I think his talk will be fascinating. Dr. Gupta has been working out of Dr. Begg's lab for her postdoc, now has her own principal lab at Brigham Women's Hospital in Boston. And she's one of the foundational people with the Kelch protein. She was actually able to discover that gene causing Nemaline myopathy herself. Dr. Gonorazky is coming to us from Canada, works side by side with Jim Dowling as an academic clinician. Look forward to hearing from him today. Okay. Dr. Gupta, we're going to have, I think you take over the slides.
All right. Thank you, Mark. Let me share my screen. And all right. Can you see my slides now?
Perfect.
Yes, perfect.
All right. So thank you, Mark. And it's a pleasure to be here today with all of you. My lab works on developing therapies in Nemaline myopathy at Brigham and Women's Hospital in Boston. And today I'm going to talk about the animal models in Nemaline myopathy research. What is the need of these models and what is the availability of this model for basic and translational research? Nemaline myopathy is a rare disorder that primarily affects skeletal muscle. So the patient has skeletal muscle weakness. The disease is clinically very heterogeneous, and we see mild to severe presentations in different patients. Many genes have been discovered in Nemaline myopathy, and two of the key hallmark features of Nemaline myopathy in all the patient, independent or irrespective of what gene is found in this patient. One is limited mobility in the patient because of the muscle weakness. And second is that when we look at the muscle biopsy of any Nemaline patient under the microscope, and here's an example, we see this presence of these dark bodies, or these are protein aggregates which are toxic to the skeletal muscle, and these are called Nemaline bodies, and that's how the name came, Nemaline myopathy. So in scientific research, we use a number of different animal models, and in the past, we have used everything from bacteria to yeast, fish, mice. Some labs are using dog and monkey also, and also cell from the human patients. And today I'm going to talk about just two of the most commonly used animal models in the skeletal muscle research, zebrafish and mice. And each of the models I mentioned earlier have their advantages and disadvantages, but most of the approaches that clinical therapies that we are testing and optimizing in the lab, we are doing using fish and mice. So when we select any animal model, we ask a number of questions. Is the disease-causing gene from the human present in the animal model? And how similar is that gene to the human? So that any therapy that we develop for that specific gene can be easily translated in the humans. And most of the zebrafish gene, 3 out of 4 genes in the human are also present in the fish. And this similarity is even higher in mice, where 99% of the human genes are present in mice. And Nemaline myopathy causing gene are 60 to 99% similar in this model. So therefore, this suggests that animal model can be used very nicely for developing therapies and understanding the disease for Nemaline myopathy. Another question we ask when we use any animal model is that how similar is the tissue of interest that we are going to study between human and these animal models? So human skeletal muscle is mostly made up of 2 kinds of muscle fibers, slow muscle fibers and fast. And similar to humans, both zebrafish and mice also have slow and fast skeletal muscle fibers. And finally, one of the consideration is the kind of therapies that we see to be used in the human patients. Can those therapies be developed in these animal models? And I'm going to talk about some of this in one of my later slides. So one of the question is, why do we need so many animal models, right? So one of the reasons we need different animal models, whether fish, whether mice, or something else, is that these different models have different versatility. So different therapeutic approaches can be developed with these models. For example, zebrafish, their embryos are really tiny, 3 to 4 millimeter in size, and large number of embryos can be obtained from one mating, like few hundred embryos. So we can test a large number of drugs using zebrafish as a model. On the other hand, mice, they are closer to human in terms of their DNA and their physiology. So they are excellent model to test single therapies to see their effectiveness as well as their toxicity. And in the past, a lot of drugs have failed when they were tested in a single model system. So it is good to have a number of different model systems. For example, if we find a drug in zebrafish, it is also good to test that in a more complex organism like mice before we take it to the human clinical trials. And another important consideration to take is that even though in the community, there are different patients, Nemaline patient, actin patient, but sometimes even if you have a mutation in the same gene, it can have different clinical presentation. And this is an example of Nemaline protein. This image is from Jim Dowling's group. Nemaline is one of the biggest protein in the human genome. And depending on where the mutation is present in the protein, it can have different clinical presentation. So it is important to have the animal models with different clinical presentations available so that we can see how good is the therapy for the mild form of the disease versus the severe form of the disease. And there are different ways in which we are using this animal model. For example, zebrafish, because their embryos are so tiny, we can take a regular lab dish with multiple wells and put a bunch of embryos in each of this well. And then we can add one drug to each of the well and evaluate the effectiveness of the drug by seeing how far they are swimming. So this is a machine, automated machine that can quantify the swimming behavior in the zebrafish. And this is an example of a recent drug screen that we are doing in a rare form of Nemaline myopathy. On the left is an example where, so each of this yellow dot represent one zebrafish. And on the left is an example where we did not add any drug. And on the right are example of different drugs that we have added to this fish, and we can see how far they can swim. So zebrafish is really good in a way. We can test a large number of therapies to narrow down to a few therapies that we can then take to the mice and test those few therapies. And Dr. Granzier's lab is testing many such compounds in the Nemaline mice model, and he's going to talk about them later. Another advantage of mice is that we can test gene therapy. So gene therapy is where we can put the normal copy of the defective gene, which is absent in the patients, by packing into some friendly viruses called AAV. And once these AAV viruses go inside the body, they go to the skeletal muscle and make the missing protein. And we are testing one of these therapy for KLHL40 gene in the mice right now. And then there are CRISPR, which are very popular these days. Here, we don't have to put the whole copy of the gene back into the body, but we can go inside the cell and fix that single DNA change, which is causing the disease. And Jim Dowling's group is working on Nemaline animal model to test CRISPRs, and hopefully you'll hear from him in future. So here is a summary of all the zebrafish and mice animal models that are available to do Nemaline myopathy research in different disease subtypes. We can notice like this is one of the field where we are lucky that we have so many animal models. We have either zebrafish or mice for almost all disease subtypes. And I would like to mention a lot of this support for developing these animal models came from AFBS in past few years, because of which now we can test a number of therapies in these animal models. So in my next few slides, I'm going to keep the focus of my talk on the zebrafish, and then Henk is going to talk about some of the mice models. I'm going to show you some examples from the way in which we are using different zebrafish models to study different diseases. So this is a data from Dr. Jim Dowling's group, who could not be here as he's traveling for a meeting to London. Dr. Dowling's group is creating a number of different zebrafish models for Nemaline. As I mentioned, Nemaline is a very big protein. And in the patient, we see a lot of clinical heterogeneity. Some patients have mild presentation, some patients have very severe presentation. And while obtaining patient muscle biopsies are very important, sometimes we obtain biopsies at different time intervals, different age group, and different parts of the skeletal muscle. So they are valuable to test certain therapies, but when we are trying to see how different changes affect the muscle function, it becomes important to create different animal models so that we can look at them at the same age and same type of muscle. So these are zebrafish embryos that we are looking under the microscopes. So in reality, they are about 2 to 3 millimeter in size, but because they are these are microscopic images, so they look much bigger here. And on the left is like how they look in the normal light. And on the right is an example where we can change the setting in the microscope, and the light falls on the muscle in such a way, if the muscle is formed normally, we see this brightness in the skeletal muscle. And if there's any defect in the organization of the skeletal muscle, this brightness gets dimmer here. So we are not taking any biopsy like we have to take from mice and human patient, and that's a big advantage of zebrafish as an animal model. We can look at the same fish over several days, how the disease is developing. So what Dr. Dowling's group has done is that he has established a number of different Nemaline models. And what we see here, like patients, some of these animal models have very disorganized skeletal muscle when we compare to the normal fish. On the other hand, some mutation causes a much milder presentation, and these fish are almost indistinguishable from the control fish. And they have looked at the muscle function by seeing how far they can swim. And we can see this dotted line. In some cases, this mutant, they do not swim as much as the control fish. But in the cases where we have milder presentation, they can swim much better. They have gone inside the skeletal muscle and looked at the Nemaline protein also. So what I'm showing you is a single muscle fiber that they have isolated from each of these different zebrafish line. And the green color represent which of these fish make the Nemaline protein. On the top is a normal fish. So what we can see from here, when they are different mutation, some result in total loss of Nemaline protein. On the other hand, some patient can still make some of the Nemaline protein. Another interesting thing is that, for example, in this case, this fish is not able to make Nemaline protein. But when we look at the skeletal muscle by staining for other proteins in pink, the muscle structure is mostly preserved. But in this case, there's no Nemaline protein and the muscle is totally gone. So these models are really great because that has helped us to explain how different mutation affect the structure of the muscle and result in skeletal muscle weakness. And when we make the animal model, we also look for like some of the key hallmark of the disease, and one is the protein aggregates, which we see in the patient, and many of this disease model also show this protein aggregates on Nemaline bodies. So these are really great model, and his group is now working on developing therapies using these disease models. So Nemaline was one example where the protein is not made when there's a mutation. In ACTA1 or actin Nemaline myopathy, this is different from Nemaline in such a way that the mutant protein or the whenever we have a change, the mutant protein is still made and is present in the skeletal muscle, and then it interferes with the function of the normal protein. So Dr. Rob Bryanson Richardson group in Monash University in Australia have created a number of actin zebrafish model. What they have done there is that they have introduced the mutant human mutation into the zebrafish, and looked at the effect of this different mutation on the skeletal muscle function and structure. So on the left here, this is an example of one of the human mutation that they have put in the fish model. And we are looking inside the skeletal muscle, and this dot or rods-like structures are the Nemaline bodies. So they found that similar to humans, when they introduce this mutation in the zebrafish, it also results in the formation of Nemaline bodies. And then they looked at the function of these different zebrafish models, and what they're looking at is the total distance traveled by this fish. So the highest bar is the control fish, normal fish that travel the farthest. And when they introduce this particular mutations, they see a decrease in the swimming ability of this fish. So again, zebrafish, so this suggests that you can replicate different kind of, or recapitulate different kind of Nemaline myopathy, either the missing protein or the defective protein by modeling them in zebrafish. So here's an example of another rare form of, now I'm going to next few slides talk about some of the rare forms of Nemaline myopathy. We have created a KLHL41 zebrafish. So in humans, when the KLHL41 protein is absent, this results in a severe form of Nemaline myopathy. So in this case, as we can see here, this is a normal fish, which has a much brighter signal under the dark light than the control fish, than the mutant fish. So this suggests that the mutant fish have very disorganized skeletal muscle. So this is very easy for us when we are looking at large number of animals rather than cutting each of the animal and looking inside. This is a very good method for us to screen these animals. And we have an automated machine where we can put this fish and look at how far they are swimming. So in the blue is the normal fish, and in the red is a fish that is not making the protein now. And we can clearly see when we analyze that over a long period of time, the mutant fish, they're never able to swim to the same extent like the normal fish. This is something similar we have seen in the human patients also, that they have reduced mobility when the KLHL41 protein is missing. And the second thing that we notice in the zebrafish is that the skeletal muscle are highly disorganized. So these are nice skeletal muscle. So it's very simple. Skeletal muscle structure is very simple. We have nice actin myosin, which form the basis of most of the skeletal muscle. And that organization is completely lost here. On top of that, we also see the presence of Nemaline bodies that we see in the human patient. So again, suggesting that we can use KLHL40 deficiency as a good model system, zebrafish as a good model system to study and develop therapies. Next, I'm going to show you an example of another rare gene called KLHL40. We have also created the zebrafish lines in my group. And this fish, they look actually normal when they are younger. On the top here, we see a normal looking control or wild type sibling. And on the bottom is a fish that is missing the KLHL40 protein. But as this fish get older, they start to look very myopathic, like weak muscle and very reduced skeletal muscle mass. And we can quantify the function of this. Does that reduced muscle mass also result in weakness of muscle? By, again, looking at the swimming behavior by different assays. And here is the swimming speed of the control fish, which is much higher than the fish that do not make the KLHL40 protein. So in addition to zebrafish, my group has, and this is the only mouse slide I'm going to show, my group has also made the KLHL40 deficient mice. This mice develop Nemaline myopathy right after birth. So these mice are about 1 week old. And on the top, we can see the normal siblings that have a normal KLHL40 protein. But on the bottom, this myopathic mice does not have any KLHL40 protein. And most of these mice die within 2 or 3 weeks after birth. So what my group is doing, we are putting the whole KLHL40 gene, which is defective in this mice, and packaging them in the viruses that are, as I mentioned, they're very friendly to human. They do not cause any major side effect. And we have already tested that these kind of viruses are making good protein in the cell culture dishes. And right now we are evaluating the effect of these viruses on restoring the lifespan and the muscle function back in the mice. And finally, I want to show you an example of another model, which is a rare form of disease, which is caused by mutation in TPM3. This work is done by a very talented postdoc, Dr. Matthijs Lambert, who is working in Dr. Luke Unkel's lab in Boston Children's Hospital. Matthijs himself is a patient with TPM3 mutation, so he's very motivated to develop therapy for himself and many other patients like him. He has made TPM3 knockout fish, and this fish exhibit reduced swimming behavior compared to the control fish. And in the middle panel, we can see this is a normal looking fish on the top. And the bottom panel is the fish that do not make any TPM3 protein. And they have this facial abnormality that we also see some of the patients with Nemaline myopathy, they have a weak facial muscle, and which is very similar what we see in the zebrafish here as well. And I briefly talked about slow and fast muscle fibers in the beginning of my presentation. And that is very critical because TPM3 is one of the genes that makes protein which is only present in the slow muscle. So what Matthijs did, he looked at the effect of missing TPM3 in the fish. He looked at both fast and slow muscle. And on the top, what we are seeing, we cut through the zebrafish, and we can then look at the specific protein by staining them with different colors. So in the green, these are the, he has stained for the slow muscle fibers. So many of you who eat fish, if you cut the fish, you can see a triangle, which is red in color on the side of the fish. So those are the slow muscle fibers, and they are perfectly formed in the normal fish. But when he looked at the TPM3 fish, he found those slow muscle fiber, the triangle is almost gone here. So this is a really cool work. This shows that TPM3 is critical for maintenance of slow muscle fiber. And when we do not have TPM3, we do not make the slow muscle fiber or are unable to maintain them, resulting in the skeletal muscle weakness. So to summarize this work, researchers have developed a number of animals to understand Nemaline myopathy, and this wide range of animal models are needed to understand the disease biology and for developing different kind of therapies. I think Nemaline myopathy is one of the good fields where zebrafish and mice models are actually recapitulate the disease biology, and that's why they're wonderful model system to develop therapies in the next steps. And finally, many therapeutic development effects are in pipeline in Nemaline myopathy for a lot of different labs. So hopefully you'll be hearing about them soon. And with that, I would like to end this talk by thanking the members of my lab, our collaborator, Dr. Granzier, and also Dr. Darling and Lambert for sharing their unpublished data for this presentation. And I would like to thank all the funding agency as well, especially a Foundation Building Strength, for supporting this work over the years. So thank you very much.
Thank you, Vandana, very much. That was a terrific overview, and thank you for all your hard work. As we've talked about, before, you've been one of the most prolific producers of research for us as a foundation, and we really appreciate that. Okay, I believe Dr. Gonorazky is going to be going next.
Okay, my turn. Thank you, Mark, for the invitation. Can you see my screen?
Yes.
Okay, perfect. Thank you, everybody. I think it's my first time actually talking in this family foundation. I started my work actually with Jim Dowling as well, working in NTM and cutting a lot of muscle, mice model, and actually cell work. And now I'm actually working more on the clinical side as a staff in the Hospital of Sick Children. So I'm the program director of neuromuscular disorders there. And I have a close collaboration with Dr. Jim Dowling. And I actually have been working also in the past on the CRISPR-Cas9 project with Dr. Granzier, mice model at Jim's lab. So today, I was asked to actually go through more of the clinical side of the Nemaline myopathies in a short period of time. So it was a challenge. So hopefully you can enjoy my talk. I don't have anything to disclose. All patients have consented to share their photos and videos. So I want to talk about a little more from the basic stuff, which is presenting you the muscle fiber. And I think this is important to understand because whenever you start looking into what we call myopathies, the big two groups that you tend to actually look is in pediatric populations are dystrophies that usually produce a muscle breakdown, and eventually congenital myopathies that usually are caused by abnormalities within the contractual apparatus or abnormalities in what we call excitation contraction coupling problems in the muscle fiber. And it depends on where in the muscle fiber you have a problem, you have a different type of congenital myopathy. For example, if you look into the storing of calcium, you tend to have what we call the core myopathies. If you have abnormalities, what we call the triad, which is a specific area in the muscle, you tend to have centronuclear myopathies. And for what we are actually looking here is actually the abnormalities in what we call the thin filament or abnormalities in the sarcomere that I'm going to talk later today will produce abnormalities in the thin filaments and eventually Nemaline myopathies. So let's look into more like in detail what I talk about excitation contraction coupling. So everything starts when the motor neuron will produce a release of specific neurotransmitters that will activate and will produce a change on the electrical activity on the muscle membrane. That electrical activity will at the end will move forward, and at the end will actually produce a change or opening of specific channels over there, okay, that will produce the release of calcium to inside the muscle cell, okay? And this is what we call the triad that I was telling you about. If you look at in electron microscopy, is this small piece of organelles right there, okay? And this calcium is extremely important because actually will help in the contraction of the muscle. So what's going to happen if we look even closer to the sarcomere that actually Dr. Gupta actually already explained? This is what we call the main part of the muscle, which is these thin filaments associated with the thick filaments here in red, okay? And the calcium, along with energy, with ATP, what we call ATP, will allow the thick filaments to bind to the thin filaments over here, and that's what will produce the contraction itself. So when we talk about thin filaments, and it's basically this lot of proteins there, the main ones are actually ACTA, or actin, musculoskeletal actin, which is the mutations are related to ACTA1, are these globular molecules that can actually be created as a filament strands, as you can see here. And Nemaline is the other protein that will allow to stabilize and eventually tell sort of how long it should actually be. But there are a lot of other proteins that we're going to show you later. So any mutations in any of these or any abnormal mutation or any changes in the genetics in these proteins will produce thin filament abnormalities and eventually what we call Nemaline myopathies. And as well as Dr. Gupta mentioned, Nemaline myopathy is really well characterized by this, the presence of what we call ROTS, which is this accumulation of protein over here. The ROTS, although it's highly indicative of a Nemaline myopathy, is actually not pathogenic. It's not only for Nemaline myopathy. You can actually find it in other disorders, but whenever you find something like this, you tend to think, first of all, for Nemaline myopathies. So these lists actually have been shown. All these genes actually can produce abnormalities on the thin filament. The most frequent ones are actually ACTA1 and NEP, but as you can see, you can have a lot of these, and we actually can characterize it by the function of them. And I think it's important to understand the function of each of them, or at least in the big group, to eventually understand how we can approach in terms of the therapeutic component. But before we actually go there, we'll just go talk a little about the clinics around it. Although you have different genes, you tend to have two main phenotypes or clinical picture. One is the classical form with an early onset and slow clinical progression, and eventually a more severe form with children with severe weakness and low tone, what we call floppy infants. You might have heard about it. And a birth with difficulties breathing and swallowing, and prognosis depend on the genetic findings. Besides these two, you can actually have a more severe form, which is a severe fetal akinesia. Dr. Gupta actually showed one picture of a baby, and eventually a milder adult onset. So as you can see, the spectrum is quite wide for this disease. It depends on the gene that you like. One have a normal gene, you might have a more severe phenotype than the other. So as I was telling you, the two main proteins in the thin filaments are Nemaline and ACTA. And in terms of what we call natural history study, probably this is the one that we have the most information. And why it's so important to have actually natural history studies, because if we are going to go to any clinical trial, it's extremely important to actually know like how is the progression of the disease. Because if we are going to implement the therapeutic, we need to know how it's actually changing the natural course of the disease. That's why I want to remark Marc's comment at the beginning. If you are going for a muscle biopsy, or it's really important the families also collaborate for the natural history studies, which is extremely important to eventually develop any kind of therapeutics down the road. And this is actually a really nice work from Dr. Dowling and Kim Marburger, who's the genetic counselor in our group. And in terms of what's the severity of the ACTA1 and NEV, as you can see, Nemaline, patients with Nemaline tend to be less severe with ACTA1, and I'm saying tending because you depending again which misspelling you have in the gene or what's the abnormality you have in the gene, but it tends to be less involved eventually in NEV than ACTA1. This is another comparison related to what we call feeding, respiratory support, and eventually what's the highest motor function. As you can see, most patients in Nemaline then tend to don't have any swallowing issues, but most of the patients with ACTA1 tend to need a G-tube, basically direct feeding through the stomach. And in terms of respiratory support, high motor function is basically sort of the same. Now Nemaline has a better prognosis. So the other genes that I want to just briefly mention is what we just mentioned about what we talk about the main structure of the gene, but there are other genes that you probably know about if one of your members of the family has suffering. It's basically these genes over here. Dr. Gupta actually already talked about it. It's one thing we can talk about these genes that actually help in the stabilization and eventually sort of remodeling of the Nemaline gene. Okay, this group over here is quite important. On the other side, if you have, as you remember, I told you about actinin. Actin is a globular protein, but it actually can go into filaments. This gene over here called cofilin or CFL2 actually helps in this what we call polymerization. So basically creating these filaments from actin. So it's really important. These patients tend to be quite severe presentation. And eventually also to anchor the thin filaments to specific areas called Z-band, it's this gene over here, NIPN. So for now, we have talked about many structures are anchoring in terms of these genes, and the other side, we have polymerization and stabilizations of the gene over here. So whenever we think about strategies of how to do therapeutics, we need to start, okay, what's actually doing, how we actually can help, and eventually what's the inheritance. And I didn't actually go into inheritance models because that's a little more complicated, but I'm happy to answer any question about the types of inheritance and how that affects the therapeutics in the groups. So when we talk about therapeutic strategy, one of the possibilities is actually to upregulate another gene that has a similar function. So during gestation, the cardiac alpha-actinin, so remember muscle, what we call skeletal muscle, has a specific form of actin called musculoskeletal actin. During gestation, actually, cardiac alpha-actin is the main actin in the muscle, the skeletal muscles as well. But in the third trimester, that actually changed, and then it's actually, it changed into the musculoskeletal actin. So what about if in the adult after birth, we can actually upregulate the cardiac alpha-actinin to actually improve the clinical picture? And that's something that has been under research. I'm not going to stay too long in each of the approaches because that's too long, and then Dr. Gupta and Dr. Granzier will talk more about it. The other possibility is actually gene replacement therapy, and Dr. Gupta talked about it. One of the main challenges in terms of gene replacement therapy, as Dr. Gupta was mentioning, is the packaging. We need to pack the gene in specific virus called adeno-associated virus that has a limited capacity. So in genes like NEV, which is huge, it's impossible to put the whole gene inside that virus. So there are other strategies, and Dr. Granzier will probably talk about it. What about if we use fragments of it? And this is an elegant work from Dr. Granzier, probably you will hear more about it. Is actually packaging eventually small fragments of it. So CRISPR-Cas9, which is actually gene editing, and I think it's important to distinguish one thing. Gene replacement therapy is you're trying to put back the gene in, and gene editing is you try to modify what is actually already there just to improve how it's actually expressed. And this is the work from the CRISPR-Cas9, and this is the mice model that actually Dr. Granzier gave us. And I spent quite a few hours with these mice when I was as a fellow. As you can see, this is a normal mice moving and trying to roll. And this is what I call the rolling test. If the mice actually was able to roll, that's actually great. And as you can see, the other small mice wasn't actually moving much. And these ones, at least for us, they were dying quite fast and in our lab. So what we were doing in specific mutations in Nemaline, we were trying to trying to actually put in again the specific fragments of the gene. And this is actually transduced myotubes, so basically small muscle fibers. In red is the actinine, and in green is the Nemaline. So they co-localize. So they mean they are like, you can see how they are together. So as you can see here, there's no green, so you just see red. But when you actually like try to rescue with CRISPR-Cas9, trying to edit the genome, what we can see is the Nemaline is expressed again. So green plus red, you have the yellow. So we talk about the structure, stabilization, and anchoring of the thin filaments, but we have this other group of genes, which is called the troponin-tropomyosin complex. As I told you, and as you remember, I told you the calcium is really important. Why is calcium important? Because the calcium will bind to this group of proteins that will allow to show specific areas on the actin protein, and this will allow the thick filaments in red here to basically bind and allow the contraction. And a lot of these, so basically it works as a lock for the calcium. So a lot of these mutations can produce two things. It's actually maybe too sensitive to calcium or really low sensitive to calcium. But I'm going to focus more in the low sensitivity, which is the most frequent form of mutations. The troponin-tropomyosin complex, there are different troponins, and they're expressed, as Dr. Gupta was mentioning, depending on the muscle fiber type, or if it's actually cardiac or it's skeletal, you have different forms. And you can actually have different clinical pictures. You can have a myopathy, you can have a cardiomyopathy, which is heart condition, or what we call, or arthrogryposis, which is basically contractures, especially on the hands or distally, or you have a combination of them. So a therapeutic targeting is actually how, if we can actually regulate the calcium sensitivity. So we can use troponin activators or eventually calcium sensitizers. So in this beautiful work from Vandana group, they use basically, they look into the thin filaments and they provide, and I'm not going to go super explain too much here, but basically, as you can see in the Y is the maximum force done by the muscle filaments and the calcium. So if you put troponin and the calcium concentration and the X, so if you with basically in the black dots is the tirasemtiv, which is the troponin activator. In these cases, they were using less amount of calcium to produce the same amount of strength than the patients that have actually or the fibers that have actually the mutation. So this is another strategy that we can actually use. So just for finalizing, this is a really nice paper, recent paper published in Review for Therapeutics. And as you can see, all the genes are here. The different strategies, I didn't go through all of them because we didn't have too much time, but there's actually quite a few strategies actually going on looking into this, which is actually quite good. And it's actually fantastic that they are actually going through. And as a summary, the clinical spectrum of Nemaline myopathies are growing. There are genes that we still, I didn't include here because we still need to have the confirmation need. There is actually limited treatment options, but however, there are actually a lot of drug targets and treatment options in development. So with that, I want to thank the whole lab. You can see here, Dr. Jim Dowling, Kim Ambergy actually did the work on, lead the work on the natural history. And here, this is just a clinical work group. I'm not including all the lab, which is actually huge as well. And they have been doing a fantastic work. And thanks again to Dr. Granzier, at least for the time I was actually working with the mice. Thank you, everybody.
Dr. Gonorazky, thank you so much. That was taking us back to science class, and also, again, showing us some of the barriers to success, which basically the Nemaline presents as 12 or 13 different diseases that we're trying to solve, each one in its own individual, representation. Okay, I think we'll move right into Dr. Granzier.
Well, good morning, everybody, or afternoon for many of you. So I'm I'm at the University of Arizona. It's a nice time of the year to be in Arizona. We have very mild weather, and we got some spillover rain from sent to us from California. So it is green in the desert, and it's a really awesome time to live in Arizona. So in my lab, I'm interested in Nemaline-based models of Nemaline myopathy. And as you already heard from Hernan and Vandana, Nemaline myopathy is characterized by hypotonia, skeletal muscle weakness, muscle hypertrophy or atrophy, and Nemaline rods, some of which you can see on this electron micrograph. I don't know if I can remove, let's see, this caption, and I don't know if you see it. So maybe some of my slide is blocked. But anyway, see if I can move that around. As you already have heard, most Nemaline myopathy genes encode thin filament proteins, or proteins that regulate the formation and maintenance of thin filaments. And this slide is taken from the Finnish group, Drs. Volkan Patterson and Pielen and colleagues, that show the many genes that are causative of Nemaline myopathy. Many target to the thin filament, one of the three main myofilaments of the skeletal muscle sarcomere, or they are involved in maintaining proteostasis and maintaining the thin filament. This shows a classification of Nemaline myopathy as published by the Finnish group again. So there are severe, typical, and mild Nemaline myopathies and other forms listed on this slide. But what I would like to draw your attention to is that Nemaline is often involved in the causative gene list of the various classes of Nemaline myopathy. So then briefly, what is Nemaline? And you already heard a little bit about that. Nemaline is a filament in the skeletal muscle sarcomere. It's a long filamentous protein that wraps around the thin filament. So this shows the sarcomeric unit of muscle, Z-disc, as Hernan already mentioned, and the thin filaments. And then Nemaline is a nearly megadalton-sized protein. It's long and slender, and it wraps around the thin filament. It is localized in the groove of the actin helix. And it spans all the way from the Z-disc that anchors the thin filament to near the tip of the thin filament. It's a complex protein. It contains many small domains, actin binding domains, and 7 of these make super repeats. And there are many super repeats. The number varies according to the species. In the mouse, there are 25. So each super repeat contains actin binding domains, and they have a conserved amino acid sequence motif. The proposed functions of Nemaline include structural roles, such as regulating the Z-disc width, and also in setting the minimal length of the thin filament, which is important for force production. Also functional roles in activating the thin filament and in tuning the cross-bridge cycling kinetics. And finally, there have been roles proposed in signaling, such as hypertrophy signaling. So Nemaline, as we heard in the previous talks, is an important Nemaline myopathy disease gene. It appears to account for up to 50% of the Nemaline myopathy cases. And Nemaline mutations that typically result in the typical form of Nemaline myopathy is autosomal recessive. And we often find compound mutations, distinct mutations on the two different Nemaline allele. This shows a result that was obtained from biopsies from patients that have an exon 55 deletion, which is found in people with Ashkenazi Jews ancestry. Nemaline can be visualized on protein gels that separate the proteins according to size. Since Nemaline is very large, it's found at the top of the gel. And you can see that in control samples, the Nemaline band is fairly intense, can be easily seen, and that is much less true in biopsies from patients with Nemaline myopathy that have this exon 55 deletion. And if you quantify how much Nemaline there is relative to controls, we find that in patients there's about 80% less Nemaline. So these patients, and these are severe Nemaline myopathy patients, are Nemaline deficient. So Nemaline-based severe Nemaline myopathy, based on this work, has a large reduction in Nemaline protein. So other mouse models of Nemaline, some of the first models that were made consisted of knocking out the full Nemaline gene. This shows earlier work, and there were two models produced in two different labs, one in my collaborator's lab, Sickkids in Germany, and the other one was in Yu Chan's lab at UCSD. At the same time, they were published. So these are full global Nemaline knockouts. And as this protein gel shows, in this case, this is a Western blot that utilizes a Nemaline antibody. So in the knockout, there is no signal visible. Nemaline is virtually absent. In the global Nemaline knockout, there is no Nemaline present, and these mice seem relatively okay at birth, suggesting that Nemaline is not critically important for embryonic development, but the mice fail to thrive and within days die. So Nemaline, as I mentioned, is not critical for embryonic development, but is important during postnatal development of skeletal muscle. We also made a conditional Nemaline knockout in which exon 2 was fluxed, and then using MCK3 expression system, Nemaline can be deleted starting from a few days before birth. And then Nemaline gradually goes down within weeks and reaches only a few % of the control levels subsequently. These mice die off during these first few weeks or few months of life, but about half survives with only a few % of Nemaline. So these are conditional Nemaline knockout mice. So about 50% of the mice survives to adulthood. And Nemaline is expressed for several weeks at an intermediate level, 20 to 60%, depending on the muscle type, and at very low levels in adult. And this is a more severe reduction than was seen in the exon 55 deletion patients. So these two mouse models represent a very severe case of Nemaline deficiency. Let's see, why isn't my slide forward? Okay, here we go. So these are two of the early mouse models that were made, the global knockout and the conditional knockout. This shows a list of mouse models that were made subsequent to that. It's based on a paper published by my colleague Koen Otterheim that gave a summary of mouse models available up until 2020. So these are the global knockouts. This is the conditional knockout. We made an exon 55 deletion mouse that Hernan already briefly talked about. The U-Chan lab published a mouse in which the C-terminal SH3 domain was deleted. And we made a mouse model in which exons 163 to 165 were deleted. As Hernan briefly mentioned, the exon 55 deletion mouse is a severe Nemaline myopathy model. Nemaline expression is absent. I don't know how well it visualizes on your monitor, but in control tissues, Nemaline is present as revealed by these green bands. But in the knockout, Nemaline is absent, unlike in the patients where there was still about 20% of the Nemaline present. But this knockout is a more severe model, and mice die within a few days after birth, resembling the global knockout. And recent work by Jim Dowling and colleagues and Hernan has provided evidence that in the mouse gene there is a pseudoexon that is localized between exons 54 and 55, and that contains a premature stop codon, and that in the exon 55 deletion model might initiate nonsense-mediated mRNA decay. An exon microarray analysis that we carried out about 10 years ago is consistent with that. And in response, Hernan and Jim have made a humanized version of the exon 55 locus. And deleting exon 55 in this humanized model results in a less severe phenotype and an easier to study mouse model that most likely will represent the human patients much better than the original exon 55 deletion model. So this mouse model is being studied by Hernan and Jim Dowling, and we currently have these mice available in Tucson, and we will utilize to study them and hopefully follow that up by therapeutic testing. The two final models listed on this slide are compound heterozygous models. One published and studied by a consortium of scientists. The last author is Dr. Nowak. And in this case, a missense mutation, as found in patients, was introduced on one allele and a premature stop on the second allele. And these mice represent a mild Nemaline-based Nemaline myopathy. They do have Nemaline rod bodies, and they have mild functional perturbation and no reduction in lifespan. And a model that we made consisted of crossing our exon 55, the original exon 55 mouse model, with a mouse in which a missense mutation was introduced in a actin binding domain that is known to enhance actin binding. And this missense mutation is a founder mutation with worldwide occurrence. These mice have a normal amount of Nemaline. They have a force deficit. They increase expression of slow myosin, slow muscle fibers, and their tropomyosin and troponin behavior is altered. And this paper was published several years ago. So having these Nemaline-based Nemaline myopathy mouse models available then allows us to perform therapeutic testing in these models. And I just want to share some summary results of this work. So part of my slide is blocked. I hope that that is not the case for you, but we started to use the calcium sensitizer that Hernan just spoke about, tirasemtiv, which is a troponin binding small molecule that slows down calcium release in multiple muscle types, but this shows results in the diaphragm. These are intact live diaphragm strips that were studied. These are wild type muscle strips, and these are strips from the conditional Nemaline knockout. I hope you can see the caption of this slide. This is work in the conditional Nemaline knockout, which can be viewed as a very severe type of Nemaline myopathy. And force production, and force is here shown as a function of the stimulation frequency. So these diaphragm strips are held in a physiological solution, and then using electrodes, they're stimulated. The range of frequencies from 1 Hertz to over 100 Hertz, and then that developed force is expressed here as a function of frequency. So you can see that in the conditional Nemaline knockout, these diaphragm muscle strips produce much less force, particularly at the low to intermediate frequencies. Force production is on the order of 20% of controls. But in the presence of tirasemtiv, and my slide is in advance, in the presence of tirasemtiv, the conditional knockout diaphragm strips produce clearly increased levels of force, and particularly at intermediate stimulation frequencies, which are likely the most important physiological frequencies. There's quite a bit of force, as this graph shows. So this shows the force increase in the presence of tirasemtiv in the diaphragm strips from the Nemaline knockout, conditional knockout mice, and particularly at intermediate frequencies, which is likely the physiological range. Force is increased on the order of 300, or the force is about 300% of the control level, so it is highly elevated. So the force is reduced in the Nemaline conditional knockout muscle, a model of severe Nemaline myopathy. And this reduction is the largest at low to intermediate stimulation frequencies, which is likely the physiological range. And then this calcium sensitizer tirasemtiv greatly increases force at these submaximal activation levels. And this is a brief summary of work that we published several years ago in this paper cited here at the bottom of the slide. And my slide again has difficulty in advancing, but let's continue to try. Okay, here we go. So the second study that utilized Nemaline-based mouse models for therapeutic testing as whether we can induce hypertrophy to improve muscle function. And in this case, we used the recently obtained compound heterozygous model and used a myostatin inhibitor. Myostatin is a negative regulator of muscle hypertrophy, and when this regulator is inactivated, it results in hypertrophy. It is a highly conserved mechanism in mammalian species. So if myostatin is inhibited, and that can be done with antibodies, for example, it results in relief of the inhibition of the AKT-mTOR pathway, which results in protein synthesis and hypertrophy. You might ask, why are we interested in it, since the work by Mike Lawler showed that myostatin inhibition does not produce weight gain or strength gain in Nemaline myopathy. But in this particular study, the conditional Nemaline knockout mouse was used, which is a severe Nemaline myopathy model. And it is possible that the mouse is simply too compromised. It has fallen off a cliff, and it is impossible to recover from this state. So we asked the question, I hope you can see this at the bottom. We hypothesized that myostatin inhibition in muscle increases protein synthesis and increases muscle size and strength in typical Nemaline myopathy. So in contrast to the earlier work that used this severe Nemaline myopathy model, a conditional knockout, in this work, we decided to, if my slides cooperate, I don't know why you have to try several times to advance the slide. Okay, here we go. We decided to use this compound head model in which this missense mutation was combined with the exon 55 model. So the exon 55 deletion removes a actin binding domain in superrepeat 9, and the 106 knock-in makes one of the actin repeats in superrepeat 22 bind stronger to actin, and making these compound head mice results in 4 genotypes, and 3 of which are tested here at the bottom in a paper that we published several years ago. And this shows that these compound heterozygous model, as well as the homozygous knock-in mice, that they have compromised force production, not so much at early age, but as these mice age in both male and females, there are significant reductions in force production. There is a normal Nemaline content in these mice. There's no excess mortality, and there is approximately a 20% reduction in muscle mass and a corresponding loss in specific force. The thin filament length is maintained, consistent with the normal Nemaline content, and there is mild fiber type switching and there are rod bodies. So this can be viewed as a model of very mild or typical Nemaline myopathy. So we asked, does myostatin inhibition improve muscle function in this mild Nemaline-based Nemaline myopathy model? And I'm trying to forward the slides. Okay, here we go. So we used an IP injection of myostatin inhibitor once per week, started at an early age, and then injected weekly until 4 months of age. And we followed the protocol published by Mike Lawler in the conditional knockout, where there was no mass and function gain. So what is the effect in this mild model? Here we go. So we applied this myostatin inhibitor, and as expected, wild-type mice have an increased muscle mass and an increased cross-sectional area. This shows results for the EDL muscle, and the compound heterozygous, which when injected with vehicle, has a reduction in muscle size, recovers significantly, almost to the level of wild type. And the absolute tetanic force level is increased as well in the compound head injected with myostatin inhibitor. If the absolute force is converted to specific force by dividing it by the cross-sectional area of the muscle, we can see that the specific force is unaffected by myostatin, which suggests that the quality of muscle that is added is the same as in the compound head treated with vehicle, but we have more muscle and therefore more force. So we conclude that in this mild Nemaline myopathy mouse model, inhibition of myostatin increases muscle size and force. And this is part of a manuscript that Johan Lindqvist, who's working on this project, is currently preparing. Apologize for my slides that have difficulty in advancing. Here we go. So then a summary is that currently in the community, there's a wide range of mouse models of Nemaline-based Nemaline myopathy available. Some represent severe Nemaline myopathy, the exon 55 model as originally made in the mouse, but that has to be restudied in the humanized mouse model that Hernan and Jim have made. But others are typical or mild forms of Nemaline myopathy. And these models are useful for therapeutic testing. And we have obtained evidence that calcium sensitizers, they enhance force at physiological activation levels. And our recent findings in the compound head model, they support that myostatin inhibition is beneficial in mild to typical Nemaline myopathy, in that there is an increased muscle mass, an increased cross-sectional area of muscle, and enhanced total force. So then acknowledgement slide. Now Johan Lindqvist has been carrying several of the projects that I touched on, and earlier work on the myostatin on terasemtiv was performed by Unjung Lee. Other members that were important in this work are listed. And then I've been fortunate to have many excellent collaborators, Jim Dowling and Hernan and others, Vandana, Mark Lawler, Koen Otterheim. Apologies for misspelling his name. I see. Yusin de Winter, Siegwille Bate, Tom Irving, and all other past, present, and future collaborators. And importantly, a lot of this work would not be possible without the support from AFBS. Not only made it possible making mouse models and doing work, some of which are represented, but it also has allowed us to leverage this funding up and get NIH funding for our work. So thank you very much for AFBS for all the support that we received throughout the years. And thank you for listening to my talk.
Yes, thank you very much. If you you can stop sharing screen now.
Okay.
Great. And it seems like, it's wonderful that the researchers had so much to share. It made our agenda seem a bit we probably overestimated how much we could cover today. So I think in general terms, what we'd like to do, instead of moving to breakout rooms, given that we've already been on for an hour and 15 minutes, I think that's probably an opportunity to list questions in the chat, or we can open up for people who want to ask questions of the researchers directly. Anyone that has questions, we know we're hoping to end by 30 after the hour, so about less than 15 minutes. but I'm happy to stay on and receive questions if people want to stay on. I can always get them to the researchers later for anyone that has to, go off and enjoy their Saturday. So in general terms, feel free to start listing any questions that you have into the chat. You can see that these scientists have dedicated a good portion of their entire work over the last years to Nemaline myopathy, and I'm really very, very grateful for their work. I see the first question coming in. Is anything for TNNT1 gene?
I don't know, maybe Vandana or Hernan knows.
I think there was a mouse model, and some of the work has been done in zebrafish models as well to model human patients' mutations there, to look at the effect of those mutations in the zebrafish by Dr. Alan Beck's group. So yes, TNT1 is one of the genes which is in our mind, and we are currently testing in both mice and zebrafish.
I believe that there is also a mice model with the gene replacement therapy data. I might be wrong, but I'm pretty sure there's.
Do you happen to know which lab tends to focus on TNNT1?
This could be JP Yen, who moved to University of Illinois at Chicago, UIC.
Mm-hmm. Got it.
Good thing about the troponin gene is it's relatively small, so I think gene therapy should be possible.
And
One
we
thing,
have a few other scientists. Go ahead, Hernan, go ahead.
yeah. So one thing in terms of TNNT1, I mean, there are different. I mean, I think some of these strategies can actually be like implemented in most of the Nemaline, like the myostatin inhibitors. I think it potentially can actually be given to the TNNT1. So some of these strategies can be broad for all of them. I mean, it depends how we see it. Gene replacement therapy, of course, has to be eventually specific for each gene. But yeah, I'm pretty sure there is a mice model published already with gene replacement.
Thank you. I want to go to Jeff Stern's question about, do we know the composition of the Nemaline body proteins? I think Jeff is a scientist in his own right and being introduced a little bit more to Nemaline today. So anyone want to manage what that composition is?
Yeah, I think scientists have looked up at the composition of these proteins, and most of these proteins are the proteins like actinin, actin, titin. These are the proteins that should be present on the main sarcomere, healthy sarcomere. But instead of staying there, they come off the sarcomere and accumulate within the skeletal muscle. So because of that, the muscle gets weaker. Does that answer that?
Yeah. Mm-hmm.
Yeah.
I think he's asking for the molecular composition.
Yeah.
Sorry.
What proteins are in these bodies? That was the question, really. What are they made of? I mean, they're very clear, big, rod-like structures. I'm sure there's some normal muscle proteins in there, but I think there has to be abnormal muscle protein in there as well. And I was wondering if anyone's done the proteome, have looked at the detailed sequence of the proteins in the bodies.
Yeah, as Vandana mentioned, these are the proteins from the sarcomere. So ACTA1 and some other of those proteins that are abnormal and that are accumulated in those Nemaline rods.
And there must be abnormal protein too, right?
Right.
Yeah.
So,
Go ahead.
What happens in some of the form like Nemaline myopathy, there's no Nemaline protein in the muscle. So in the absence of Nemaline, still many proteins from the sarcomere fall off and forms this abnormal protein body. So those proteins are not available now to make healthy muscle. And in disease like actin Nemaline myopathy, where the abnormal protein is still there in the muscle, that also comes and forms a part of the Nemaline bodies. So depending on which type of Nemaline myopathy you have, some of the proteins composition may change, some of the abnormal protein as well as normal protein.
And thank you all for your, the time and presentations. It's very educational. Very grateful. Thank you.
Welcome.
Thank you. Gus, do you mind taking on Kevin's question, which talks about pluripotent stem cells, and talk a bit about our models with Dr. Mack right now?
Yeah, this was actually my question. And I am also a scientist. Jeff and I are married. And I do a lot of induced pluripotent stem cell modeling for neurodegenerative disease. And the models you describe are beautiful. But I wondered if there was a place for iPSC-derived muscles as an additional in vitro model.
Exactly, as Marc mentioned, we AFBS was funding in the last 4 years studies on iPSCs. A group in Australia had established 4 new lines of iPSCs for ACTA1. In this case, there are, of course, other groups working on other Nemaline proteins. And one thing that in the case of the ACTA1 is that with the iPSCs, they were not able to fully have fully mature cells. So now what we are evolving to is a next step that is another group that is collaborating with the Australian group that is doing 3D muscles. So using those iPS cells, making a matrix where those cells can differentiate to more mature phenotypes, and also the nice thing of this new 3D model is that you can measure force and other contractile properties of the in vitro model, the muscle in vitro model. So yes, there is a lot of work going on in that, and yeah, people around the world is doing 3D models for also the heart in cases of muscle pathologies where heart is involved, etc.
Thank you.
Yeah, the nice thing is that can be done in, let's say, a 24-well dish, and so you have many engineered skeletal muscle tissues that can then be used for drug testing, for example. We do this for engineered heart tissues, where we make them from human iPSC-derived cardiac myocytes. But I know that this work is going on for skeletal muscle, and I think that potentially has a lot of power, high throughput, and then also personalized medicine.
Thank you.
Dr. Gonorazky, do you mind taking Rebecca's question about why do familial NM patients with the exact same mutation present differently phenotypically?
Yeah, because it might be modifiers as well. And it depends on the mutations. For example, if you talk about ACTA1, I mean, first we need to differentiate the inheritance pattern, which is, for example, dominant versus recessive mutations. So in dominant mutations, if you have 2 copies of the gene, one from mom, one from dad, dominant mutations need to have, for example, one abnormal and then the other one is normal. In recessive mutations, you need to have both 2 abnormal copies to have the disease. Dominant mutations can actually have a huge variability on the disease, on what we call penetrance. For you to just simplify, understand, if you have 2 copies of or you have 2 cars, but one is like a really bad car and it's not working properly, but the other one is like a Ferrari and doing fantastic, it will take you where you need to go. But if you have one that it's like an average car, but the other one is actually really abnormal, then it will take you, but not as fast or not as good. It's sort of like that. So it depends on which gene you are talking about, and again, on the inheritance pattern as well. Hope that was clear.
Rebecca, I'm going to allow you to ask your question maybe a little more detailed, because I know you're a scientist as well.
Hi, thank you. Thank you so much for all of your talks, and it was helpful. I kept noticing a common theme is that the mutation dictates the phenotype, but also at the same time, on like the clinical end, I see that people with the same exact dominant mutation do have different outcomes, like sometimes drastically. So I guess ACTA1 would be a way to control for that. But on the dominant, the dominant mutations, what do you think is affecting each person's outcome, or within a family? Why are two people who have the same dominant mutation exhibiting the disease differently?
So
okay.
Sorry, let's.
No, go ahead.
I mean, one of the possibilities is actually, did you see the remember that I talked about the cardiac actinin? It is a possibility that there are family members that have actually higher expression of the cardiac actinin in the muscle, causing that the disease is less severe, but it might be more modifiers. I don't know, Gus, you were going to say anything else?
Yeah, I was going to, especially, I didn't know, but Rebecca, you are a scientist. You know that we always have these problems with the genetic background. And usually when you work in the lab with these animal models, you have a purified background. So all the other genes are the same in all the mice, for example, but you have the mutant NEB or the mutant ACTA. In the case of humans, we cannot control that. And all the gene variants of each individual that are different created a different penetrance of the mutation, and that's a big probably that's the current hypothesis of why there is this variability in phenotypic expression because of the genetic background of each individual that is different. The combination that coming from your father and mother and your ancestors, all that variability of genes may generate, and that's a source also of research to find which are these probably modifier genes that you can target to. If you find one individual that is very mild affected with another with the same mutation with a very bad progression of the disease, something in that individual that is mildly affected is good to modulate or to target. So there is a big search of these genetic modifiers to try to use it as targets for therapeutics.
That makes sense. Thank you so much.
In general terms, I know it is 30 after the hour, so anyone who would like to drop off, certainly understanding. We're going to continue to record, stay on for a few more minutes. There's a couple of maybe brief questions.
And I would like to add that probably our next webinar, we will do this breakout sessions, and so we can focus on different genes.
Yeah, so we'll pull together the breakout sessions without some of the research lectures to really give the opportunity for the people with different subtypes to be able to communicate and actually meet a few people that actually have their own subtype, so they could actually compare notes a little bit and give some voice to those people to chat and connect. And we'll share whatever different symptoms you may be experiencing and what physical attributes you might have with the researchers, so they know as well. And we can share a little bit more in that vein. There was a question about how would the mechanism of a therapy be derived, and would it be delivered? Is it would it be a pill, or would it be an IV or an injection like a flu shot carrying a virus? Gus, do you want to take that one?
Yes, well, all those modalities, depending on, it will be specific for the type of product that you will use as medicine. In the case of gene therapy, you will need most probably to inject IV, although there are some potential uses in localized muscle injections. But of course, some other like the tirasemib, that is you can have it in a pill and it be administered orally. So yeah, I mean, it would depend. All those possibilities, all those administration ways will be possible depending on the medicine.
I think I can answer Jamie Stanley's question about supplements. Really, there haven't been any validated supplements to really support any of the Nemaline myopathy subtypes. There was some case studies back ways about tyrosine, and there were a few back in the 80s even published papers by a PhD family that actually had some scientific background, obviously, but they wrote a case paper on tyrosine. So tyrosine has been written up to be to do two things primarily, maybe boost energy, but also dry secretions. Those are the two case papers that came out. And again, it hasn't been validated. And I think we've even tested it in some of our zebrafish models, and they've not shown any increased mobility or motility in those actions. So, but again, I think it's definitely benign enough to consider trying if you haven't tried anything like that. Obviously, healthy diets and an activity level that sort of at least maintains current function and maybe stresses the body a little bit to be able to make sure that you're not going to move into a decline is important. Obviously, balancing that with the daily tasks of all the different actions that you have to carry out. But we really hope that we can allow people to get a little bit of stress, appropriate stress, to be able to kind of develop muscle wherever possible. Tova Cooper had a question. There may also not be known. Is there a hypothesis for what the outcome of gene replacement, gene editing would be? After receiving one of these treatments, how would a person clinically present? A moderate form of NM, for example.
I can talk.
Yeah.
Sorry, Dr. Gupta, please.
Yes. So the outcome can be pretty different in different patients. In the severe form, where the lifespan is short, gene therapy has been shown to increase a lifespan tremendously by several times. And in the cases when the disease is still moderate, adding a virus back in a disease like Nemaline myopathy, we predict to increase the size of the muscle fiber diameter, which is small, will increase, and hopefully it will also increase the force production by the muscle. So it will increase overall muscle performance and mobility. So Hernan, do you want to add to that?
No, you have more experience than me. I mean, I have experience from other gene replacement therapies, NTN, DMD, and SMA. I mean, as you said, like the SMA population, now, thanks to newborn screening, if we intervene soon enough, the kids are doing fantastic. But again, I mean, it depends on the disease, how they're going to progress, what's the underlying condition. In MTM, for example, which is the other congenital myopathy, there were serious complications on the liver because those patients actually have a background liver involvement. And usually what you're going to do with gene replacement therapy, most likely you're going to inject through IV, and the liver is the main filter. We will capture most of it and it will have the major hit. So I guess it will really depend. In MTN, I mean, except those patients, the patients are doing really well, to tell the truth. And that's a congenital myopathy.
Thank you. And Henk, do you mind taking Laura Liu's question? Her child has one NEB mutation at splicing site. Does it mean that NEB protein level is downregulated?
I think it could be, or there could be changes in splicing. I think it's hard to predict. I think it would have to be known. One would have to look at carefully where the mutation is. I'm not a well positioned to make a prediction, but I think somebody should be able to take a close look at where that mutation is, and if that could cause changes in exon inclusion, or what might happen. I think if Jim would be here, he probably would have a better answer, or maybe Hernan has.
No, I agree with you. I mean, it's difficult to know. Usually, I mean, NEB, you have two mutations, no? And in terms of the splicing, we have studied splicing a lot, but it depends where you have the splicing and what's actually causing. It can produce an intronic inclusion, producing like a premature stop codon, or just the domain effect, like basically changing the whole structure of the protein. It can produce skipping, basically a skipping of the next exon. So it really depends on what's going on there. And eventually the combination with the second mutation that I'm not sure if your kid has it.
And I think it is important in those cases to contribute a biopsy to some of the researchers, because in the past we have studied biopsy from patient with these kind of questions where a mutation does it affect the Nemaline level or not? So we have worked with those biopsy. We have sent some of the sample to Dr. Granzier's lab also, where they have looked at the Western blot, the data he showed, and they concluded whether a mutation is affecting the level of Nemaline, because sometimes it's just hard to predict by looking at the change in front of you. You have to do additional experiments.
Yeah, I think that is definitely the case. So having a muscle biopsy available would allow us to learn a lot about the size of the protein, if certain areas are missing, and if the expression level is similar to normal. So it would be really valuable to get a muscle biopsy from this patient. It wouldn't take a whole lot of tissue.
Mhm. Thank you. So Laura, if we want to have follow-up or introduction on the side, let me know if you have a biopsy available that might be shared. Verena Farr asked, Gus, I don't know if you want to take this one. Are we aware of any instances where the fetal cardiac alpha actin is compromised or mutated in combination with compromised skeletal actin, resulting in a more severe form?
I think as Hernan, I think answer in the chat that is an interesting question. It's a very interesting question, and he said about some publishing data soon. I don't know. I didn't know about that.
Or I can't disclose, but interesting. We actually found some patients with the actin ACTC1 changes, but we'll be publishing that group soon.
Okay, so as soon as it's published, we will come back to this and answer better that question.
Sharon Smith's asking about creatine. I don't know that creatine's ever been in the drug samplings or support in our zebrafish. Was that anything you tested, Dr. Gupta?
No, we have not tested creatine so far. That's an interesting question, because sometimes we do see some changes in the mitochondria. So that would be something to look into in the near future. Thank you.
And I would think on the milder forms, Sharon, that creatine could have benefit when the muscle structure of the protein itself is highly irregular. Then the impact of the creatine may not have as much impact. So the more typical or more mild types would probably see some benefit from, again, just energy production in the muscle. Jeff, do you want to go ahead and ask your question?
Well, it's a simple-minded question that we see all these muscle builders out there building up their muscles to extraordinary amounts. I guess the myostatin inhibitors caught my interest because I believe they're used by muscle builders. I don't know if that's the right word. The people, bodybuilders. Thank you. And there are a number of drugs that are used by these bodybuilders. This show Sally we saw on Netflix. The woman was only taking 3 of them, but the husband was taking 5. Have you looked at, Henk? Have you looked at these?
No, I haven't. This inhibitor that we tested definitely works very well on wild type mice also. So it would work on and presumably would work on humans as well, but I don't know what bodybuilders all use, and there are definitely side effects also that one has to be very careful with. So I would not take anything off the shelf.
Oh yeah, of course. No, I'm just curious in the animal models, not.
Yeah, I'm sure that this work on that, but I'm not up on that.
Got it.
And there is also always this discussion that maybe even in the case of Nemaline myopathy, that the problem is in the minimal in the mechanical base of the muscle, that so the contractile apparatus. Even if you increase the mass of the muscle, you will not fix the contractile dysfunction. So probably you even do even worse if you have a big muscle with a contractile apparatus that is not working, because the force it will be even reduced.
But our data suggests that the specific force is not affected, so it doesn't. You don't get more worse muscle. You get just more of the same, which is still beneficial. You have more total force, but the quality of the muscle has not improved. But it hasn't gotten worse either with this particular inhibitor, at least. So I think that is good news. In that you should have more function.
Thank you, everyone. I think I want to try and wrap with the two last questions. Natalia asks, said, my daughter was identified NEB compound heterozygous state mutation. How is this case illness will progress? Is it non-standard variant of the disease? Can anyone take a venture at what that?
So it really depends on the point mutations that you have. That's like we need to look into that. In general, as I mentioned, Nemaline tends to be like congenital myopathies in general tend to be slowly progressive or sort of stable. So again, it depends not only in the gene, but where and how where it's located in the gene and how it's actually impacting the protein. So it's extremely difficult to like no, just at that. I mean, yeah.
Yeah, the location would be very important. If just a small piece is missing, it might be a mild, or the disease might not be progressive, but it all depends on the fine details where the mutations are.
Okay, and I think just to finalize, last two questions, can an old biopsy be taken from the calf, still be looked at 17 years later? And I think the scientists would probably say if the storage,
yeah, it depends on the storage. If it was stored in a -80 freezer, it definitely would be fine to look at. So it all depends on how it was taken. That is important. Ideally, very quickly frozen in liquid nitrogen. And stored at -80 or lower temperatures, degrees Celsius.
And then Surya's question was about taurine supplements. Again, no information on taurine. I don't believe that we used it in our zebrafish baths. That I think we used 1,500 different compounds, but I don't believe taurine was one of them. I think they were specifically FDA controlled drugs. Is that right?
I think Rob Bryson Richardson had tested that in Nemaline model at one time, and he did not see any improvement in muscle function or performance.
Okay. All right. Well, I just want to say thank you to the high attendance today. Really appreciate everyone listening in. We had a lot of people stay on the entire time. The scientists who have made themselves available on their Saturdays to really take time to share their amazing research. And for our team that's here, I just want to make sure if anyone hasn't checked out our new website, new logo, new support and resource pages for the community. I want to thank Jen Tozer, who's not here today, but she's helped been critical in getting that out there. And if anyone is interested, there's information there about our weekend of building strength. I'm wearing the old shirt from last year. Just to say that on April 29th and 30th, we're trying to host a bunch of fundraisers. And so around the world, hoping that people will step up and do a garage sale or a little mini walk with their friends to be able to help raise some money for the foundation. More details on our website. Is there any final thoughts or questions from our team? Okay, great. And please, everybody, enjoy your Saturday. Let us know if you have any follow-up questions. Please reach out anytime.
And thank you, AFBS.
Thanks, Henk. Yeah, thank you.
Thank you, Henk. Thank you, Hernan. Thank you, Vandana.